Hello, how are you guys doing? So let's go ahead and get started. Let's see. I'll just write down. Okay. It is pie day. Whoopee. Yeah, go to Marie Callender's, get some pie. Let's see. All right. So before we get into a deep dive, are there any questions you guys have? Anything you guys were maybe reviewing, saw, and had any questions about? What do you guys think? Of course, my prayer doesn't want to exist correctly. Any questions at all? Okay. So did you guys get a chance to look at that outline I sent through Remind? Yeah. Do you think that was helpful?
Yes, it was, but I think the quiz was too easy.
It was too easy.
Yeah, there was a lot of mostly just true and false.
Mm-hmm. That's really what this quiz was. It wasn't too tricky because it was a lot more memorization rather than really
Yeah,
complex things. Yeah.
yeah, there was no trick questions.
Mm-hmm. But yeah. So that was always a good studying habit of mine, was to just take the PowerPoints and try to outline them like that. And if I didn't really understand something, I would elaborate. And if I understood something, I would just include it in the outline. I would just, if it was like something, let's say for a certain test, I knew what the reagents were, I just didn't know what they did, then I would go into depth and be like, okay, this is the test, these are the reagents, this is what they do. But if I knew the reagents and what they did, and like I could do that off the top of my head, I would just put the test and the reagent names, and that was it. So yeah, that's just how I studied. Um, and I thought I'd show an example because it's a little hard to just describe. Um, but yeah, so I'm glad you guys found that helpful. Let's see. So yeah, it's not too bad of a quiz. Were there any that were like you were on the cusp, like you were like, oh, this one might be just a little bit harder, or were they all just like, man, I know this?
Well, the one version I had asked all of the stuff that goes into the PCR.
Mm-hmm.
And there was one I was like, wait, are there primers? Are there not primers? But I
Mm-hmm.
knew there was primers, so I was just not confident, but I got the right answer now that I looked at it.
Good, good. But yeah. So let's see what we're gonna go over. So there was a lot of work on the unknown today. Some of you guys found out what you guys got already. But I did want to show you guys, here, let me actually pull it up before I start talking about it. I wanted to show you guys my unknown, and I actually have two versions. Let's see if I can find them. So it's gonna be a ways back. Hmm. Hmm. Man. Okay, let's try this. Because Dr. B was talking about how she wanted me to show you guys not only dang, not only did she want me to show you guys the of my end product that got 100%, but also my in between. So I'm just trying to find it. I don't have no idea where it is. Oh, okay. Here we go. Will it open? Yes. Score. And then I'll find my end. Probably should have done this earlier, but here we go. All right. So I'm going to first share my screen with my initial project. I'll use it here. Okay. So my initial, let's see, how many pages was it? It was 19 pages, including the works. So my initial was 19 pages, and she because it was also really indented, so it was probably less than that in actuality, but it was I had like an error here about formatting, and the big thing is that you need to use complete sentences. Because it was just like symptoms versus signs, so I was like, I just broke it up basically. But let's see. But like here it says look up stats on percentage of patients that have cancer and this infection. So if they wanted, she wanted some statistics, and then look at all these edits in the margins, right? It was a little bit of a beast. Further development, right? She wants a lot of detail. Like I can say, she was diagnosed with this leukemia, and her age and the fact that she's living in this nursing facility could have an impact on her emotional health. I needed to say that what feelings were being felt, how it could impact her, et cetera, et cetera. I need to develop that more. Another development. Another thing, do not capitalize your second thing. It always is it was like on all of these, like staph epidermidis, the E is supposed to be lowercase. Like that was my biggest thing all year. I do not know why. And then she was like, what percentage of cancer patients with a similar profile as your patient have had this type of infection? And then from that data, I was supposed to talk about everything else, right? It was a holistic view. Let's see. So yes, so like she wanted more information here. No quotes whatsoever, nothing. No quotes throughout this entire thing. This was good for her, and I just needed to fix my nomenclature, and then I also needed to put this in a sentence. So I just needed to say that E. coli is a gram-negative, broad oxidase negative, superlactose fermenter, indole positive, citrate negative, and those are the properties, right? But I needed to put it in full sentences. I needed more details on these microbes. It was a whole lot of detail. And then also, this is what you guys had today, or tomorrow you'll have it, about your Streakster isolation. So one thing that it was like, apparent that we need a lot more detail than what I was giving. It's because this is a TSA plate. It's a nutrient agar. It would allow me to see plain growth and colony morphology, and I would be able to take another sample for test. This was not a good enough amount of detail. She wanted what was a TSA plate made of and what makes an enrichment. And then I also needed to, she's been stressing the whole mixed colony thing. I needed to state that there could be a mixed colony and that I took this and this is what I had. So one thing here was that I could not get any isolates on my TSA because I had a special type of cell. It's a swarming bacteria, which means that it just covered the whole plate. It basically created one big film. So it was pretty freaky. So I couldn't even sea best it, but you would sea best it. Let's see. Yes. So this is it right here. You should be able to see through it pretty well. And so I put a reference picture here. But yeah, let's see. And yeah, it was just, I had like an F. I really had a hard time doing the E and B because it wasn't so like enriched. It almost looked brown. It was weird. And then I also said, it would be purple, but it can be purple, pink, or green if it's positive. Yeah, look, my E and B was like brown. Like it was weird. But she said that they're apparently like translucent or something or other. I don't know. But I just interpreted the results that it did not ferment lactose. And then the next thing is a gram stain to discover the morphology of the bacteria. I had a negative bacillus. And like as I was doing these tests, I was able to rule out some of my differentials. So like I had E. coli, right? Because this didn't ferment lactose. I knew I didn't have E. coli, so I knocked it off. So with this, I actually was able to get down pretty close to what I had. But with this gram stain piece up here, you need to say like, if it's gram positive, it'll be this. If it's gram negative, it'll be this. Now, since mine was gram negative, that means its cell wall looks like this, right? And then you can't just say, oh, I had a gram negative. Just say, I discovered that under the microscope, it appeared purple with rod shaped bacteria. So I have a gram negative, or I'm sorry, a gram positive rod, right? In my case, I had a gram negative, but you kind of need to like talk about what it looks like, embed all of these types of pictures. And then Bergey's keys, stress it. You could say like, instead of saying like, in order to fix it here, and I would say, in order to figure out what test to do next, I would consult Bergey's keys. Bergey's keys tells me that the next test I need to perform is an oxidase test. So that's what I would do. Let's see. What is the oxidase test for? What does the results mean? You really want to like talk about the theory behind every single one of the tests. Let's see. Okay, so you know how on your lab manual it says indole, it says citrate, it has like all of those things. Briefly look into those things and say, you know, what tests it's testing for and what it means by positive or negative. Let's see. And then also discuss some pros and cons of the microgen, right? I had another non-proper nomenclature, but we don't talk about that. This was the big Kona. So like the comorbidities for me, I was like, yeah, she's immunocompromised, like really bad. But I had to talk about why. I had to go a little bit more in depth into the whole chronic lymphostatic leukemia. I had to talk about how that affect her immune system in general, and then on top of that, having a bacterial infection. Not only that, but she was undergoing chemo. What does chemo do to your immune system? How does that impact your how prone you are to getting an infection? And then I had to include statistics, lots of numbers. You'll learn this later. The flow chart comes a lot later, but yeah, and you'll figure out how to do it step by step. You're fine. But you need to discuss that said flow chart in complete sentences. But yes. This was just me being like, I don't think I did anything here. I think I just forgot to do it. Let's see. You kind of have to think about like what caused it. Like if you had a sore, then like let's say it's a cut while doing slicing onions or something, right? Then it would be, you know, the patient cut her fingertip off, which means she needed to get stitches and yada yada yada, right? So you would have to discuss things like that. Mine was specifically a pararectal abscess, which meant that it could be from a bed sore. If they weren't being turned properly, then all of a sudden they had this pressure injury that opened up. And when it opened up, it offered a portal for the bacteria to enter and cause an infection. So I needed to develop that a lot more. She was also put on laxatives for some reason, leading up to the disease, which is like, I don't know, that was weird. And I couldn't just say, oh, I would use a catheter in this situation. I would have to say why, right? I'd have to say why I'm doing these things and how I'm going to do them, and are they going to be beneficial for the patient, right? Please go back up a little. I had to reword this because it was weird. But you do have to discuss why you chose certain antibiotics, right? And what data supports it. So if you were to have let's see, if you had a gram negative, then you would want something that targets cell wall synthesis, right? So if you have this gram negative and you said, okay, I'm gonna give it penicillin, right? Then you would have to discuss why that works. And you'd have to discuss what it targets specifically. And I also included here, I'll show you my measurements. And then I also included the dosage or the micrograms. Yes. So this is where she gave me credit. This is where I got credit. I said I'm gonna choose this antibiotic and this antibiotic, and I'm gonna give it 2 grams IV every 6 to 8 hours for severe infections with a max dosage of 12 grams a day, either intramuscularly or intravenously. So I said that, and I gave a good amount of detail, and I discussed later what these two targeted, because look, she asked right here, what does a target? So it's really in depth. A layman has to be able to read this and understand what you're talking about. But yes, also here, studies showed that 100% of 25 P. vulgaris isolates were susceptible to 4 broad spectrum cephalosporins by disc diffusion, and 40% of 64 were susceptible to both of the ones that I chose. So this is the type of data she wanted. I said something about sitz baths. So she said, what is that and why is it recommended? And then let's see. For this one, she wanted me to turn this into one number, basically. Like she needs to come back in like 2 weeks. Because she's extremely immune compromised. Like it couldn't just be so broad as to apply to anybody with this type of infection. It had to be very specific to my patient. Let's see. Oh, I also had to talk about anything other medical things that I had to treat. So let's say, she has leukemia, right? My patient has leukemia. She got a secondary infection, but she also has no family and she's living in a retirement home. She's all alone and she understands that, her roommate was already passed, right? She understands mortality and she is getting now scared, right? Like I think any of us would be like that. So you kind of have to discuss, okay, well, this person's scared and they need reassurance. They need some help for mental counseling. They need to, need to get their oncologist together with their nursing facility, and the whole team needs to talk about how to treat this patient. And maybe even the transition to home hospice. That's something like end of life care. It has to you have to choose between, are you going to fight this as aggressively as possible, or are you going to focus on comfort and rather than knocking that person out with chemo and antibiotics and just everything, right? But yeah. Let's see, what data supports her likelihood of survival? I think that one was just like, I'm trying to think exactly what she wanted me, because I asked her a few questions about this. I think she was just like, she kind of knew my patient scenario already. So she was like, you seem pretty optimistic about this. Why do you think that she's going to survive this? And I was like, I just don't want my patient to die. So I kind of had to bite the bullet and say, yeah, my patient, it does not have a good outcome. And I did talk about how we should probably change from an aggressive treatment to end of life care at a certain point, depending on because again, doctors can't make that call. It's like, I'm not going to treat you. You're just going to have to prepare to die. Doctors can't make that call. Patients have to. So part of my thing was I had to sit down and say, okay, in order to prepare this patient with this news, I'm going to sit down the patient, tell them what's going on and their diagnoses, and I'm going to ask them to think about what the next step they'd like. Here are our options. What do they want to do? If we decide to attack this, you're going to be undergoing chemo, you're going to have antibiotics, and there's a high likelihood that's not going to work. But if you do decide to undergo home hospice and end of life care, we can arrange that, right? So that was what she wanted me to focus on, rather than just saying, if she makes it, hopefully, we would do this. She wanted us to give us the options to the patient. Like I said, educate the patient on the treatment. And then also, my patient specifically was in a nursing home. So I had to also talk about how I would discuss this with the nursing team. And I would say, listen, this patient has this infection. You need to put this patient in an isolated room, or you need to pay very close attention and wash your hands thoroughly after interacting with this patient. And you need to make sure that they have a bath at least once a day, if not twice a day, right? Like things that would be different from the norm. But yeah, let's see. So like she said good here because I had a percentage. And I said that this is how we treat this, right? Yeah. This is what she said. But yeah. And then this was just how I cited mine. Mine was just MLA. So yeah, that's a lot. And when I read this, I cried because I poured my heart and soul in this initially. So it was very upsetting to see that it was not nearly good enough what it needed to be. But here we are. This is my 1 of 27. So I added a solid 8 pages on top of it. And I even, if you look at it, it's less skinny. It's a little thicker. A lot more information. So you see, I'm just gonna scroll down randomly. I should be able to find, if it's not a picture, I should be able to find a percentage. Here we go. 70%, right? And then let's go down here. This is another little area, but I added a lot of information, right? And then when I go down here, right, I said morbidity was high in the first part of my paper, right? I started off with that. I knew that information. I needed the information to back it up. I got the general idea, but I also needed to say that it has, again, a high morbidity and a recurrence rate and chronic pain in more than 30% of patients. I needed to provide evidence with my claims. Like I couldn't just say that probably hurts. Like I needed to have proof that it hurts, right? And then because of that, I added like a whole page of resources because I had to get so many more viable I had to get so many more viable statistics and I had to put them all in. And then here, I also wanted to show you. So yeah, one possible pathogen is E. coli. I fixed my nomenclature due to proximity to the anus. E. coli is a gram-negative rod, oxidase negative, a superlactose fermenter, indole positive, and citrate negative. That was what I said already. I just put in the whole sentences and I fixed my nomenclature, right? So it's kind of it's a big one. I would suggest, because look, with the information, if regardless of if you saw your plates today, with the information you have, you should be able to get to, let's see, you should get past the differential, and you should be able to talk about what tests you're going to perform, not necessarily what the results were. So you could outline this pretty well. So the first test I performed was a streak for isolation on TSA, MSA, and EMB plates. And then down here, I said, I described what they were, and then you could leave a space for results. And then you can say what you did next, and then leave a space for results, what you did next, space for results. And you should be able to get that done without getting any results from any tests. From today, if you got your plates back, then you should be able to start filling this section in and adding your pictures. Let's see. And then talk about the gram stain and your steps, right? And then insert that picture in. And then you should also be able to put in probably something about Bergey's keys, right? To find out that you should do an oxidase test. And then right after here should be like your stopping point. So talking about what the microgen test does and what the procedure is, and then stopping before you get to the whole results is good. I included this, and then if you want your own little A and B panel, like little paper like this, we have extra in the lab and you can take one. I will find one for you next time we're in lab. But you also want this paper at the end, right? You could also do this, the in-depth discussion about the patient's immune status. You could do that without even getting any tests. This is going to be after lab 18. It's going to be a while until you can do this one. So just hold off on this one. This was the last thing I even did. But you're going to do a flow chart. Let's see what else. You have to have your pathogen, note that one. This, you can probably start this a little bit. Treatment of the etiological agent. That one you do need to know what it is. And then you have to pick from your bacterias. You have to pick the antibiotics and test them on the Kirby-Bauer test, the Mueller-Hilton plate. And let's see. Emotional status, include that. You can probably do that now. And yeah, everything else you kind of need to know what the bacteria is, or you need to have the test results already. But you can start with those sections, like now. You can if you want. I would highly recommend them. I would highly recommend that, because the thing is, this and a couple other things are due on the same day, and then the micro projects due a week later. So it's like one of those things where it's a lot all at once. So if you can knock out what you can now, that'd be great. That would allow for you to have more time that you could spend on some other things. But definitely get as much done as you can now. Let me look at, I was gonna look at, let's see. Well, you know what? I'm gonna go ahead and let you guys off with a break real quick. Come back at 5:49. 5:49 was probably good. I'll probably start. Sorry, 5:49. I meant to say 39. I'll probably start at 5:40 again. But be back by 39 just in case. Okay, so go ahead and take a quick breather, and then we'll talk about case study, and I'll also talk about one other thing I was going to tell you guys about, but I need to find it. My train of thought was lost. But definitely a little bit about case study too, and then any questions you might have about labs. Okay, I'm going to cut you guys off, and we'll be back in about 5-6 minutes. Okay, peace out. See you in a second.